I suggest that we should get started. Hello and welcome to Colloquium. As a general reminder to everyone, this meeting is recorded. During the talk, we would encourage all of you to keep your microphone muted unless you're asking a question. But please feel free to keep your video feed on if your internet bandwidth allows, because the speakers always enjoy interacting with our faces rather than little black squares. If you want to ask a question, please feel free to either raise your hand or type it in the chat window, or the speaker said that he is happy to answer even if you unmute yourself and ask directly. It is my great pleasure to introduce our speaker today, Professor Doron Levy from the University of Maryland. He is current chair of the math department there, also the director of the Center for Scientific Computation and Mathematical Modeling. Doron is a Guggenheim Fellow, a Pauli Fellow, was affiliated with a number of different institutions, including Stanford University, UC Berkeley, and Lawrence Berkeley National Lab, where we have overlapped when I was a graduate student and he was a postdoc. Also the University of Paris 6 and the École Normale Supérieure. You can find all of this information on his website. What you will not find there is that he is also a winner of several contests among the magicians. So whenever he does give a talk, I watch his hands very carefully. It's always a delight. Doron will be telling us about fighting drug resistance with mathematics. So please take it away.
Thanks, Alex. A wonderful introduction. And it really is a pleasure, an honor and a pleasure to speak to you today and visit you, if only virtually. So hopefully one of these days we all go back to normal and we can do that in person. Yeah, the topic of the day is fighting drug resistance in math. So one thing that I would like to emphasize is that that's a work with a group of people, and I put several names here, and I ask forgiveness for those that I did not mention, so some former postdocs and students. But one thing that is important is that there are doctors involved, and that is something that is very characteristic to things I'm doing these days. I will say this is not going to be exactly a data-driven kind of talk. There are also these kinds of projects, and it will be mathematical at times, but what is important is to say that all the questions really are questions that are being raised by doctors, and math is in some sense nothing but a tool to address these questions. So Michael Gottesman, a collaborator of mine, he's the head of the Laboratory of Cell Biology at the National Cancer Institute in Bethesda, not far from campus, in fact halfway between where I live and campus. Michael is one of these very few doctors, still very few doctors, that think that math can actually help them figure out how to better treat patients. And he, in addition to the role as the chief of the Laboratory of Cell Biology, he is also the deputy director of the entire NIH, so he's in charge of their intramural funding. So it certainly is good to have someone so high up in the National Institute of Health chain that believes that math is useful. By the way, his son is a physicist, so it's not because of me. What I would like to tell you today about is, as promised, some drug resistance and math and how do they come together. And I'll start with a little bit of biology there just to give you a feeling. The main message is like, this is an enormously complex problem. So forget about ever being able to do anything accurately here, but nevertheless, one can slice some pieces of problems that can be interesting, both from the application perspective and the math. And I'll try to give you a taste about that. Now, one thing that is important to mention is mathematics and drug resistance did not start with cancer studies, so that will give me a good opportunity to mention something that is culturally interesting, and we will get there. So multiple drug resistance, this is a caricature, a cartoon of a cell taken from a paper by Gillian Gottesman about a decade ago. What you see here, you do see the, as I move the yeah, everyone's good. Okay, good. So you see here a cell. What you have around the cell is a membrane of the cell. Here's the nucleus of the cell. The DNA sits inside of the nucleus. And when you administer a drug, the drug can effectively do many different things. But let's say that you want the drug to penetrate into the cell, then penetrate into the nucleus, damage the DNA, and prevent that cell from replicating. Now, I'm not getting into the questions of how does a drug know that this cell is its target? And in many cases, it does not. So, you know, along the way, you kill cells you're not supposed to. But there are many mechanisms that prevent drugs from doing what they're supposed to do. So let me try to slightly parse this picture. One thing that happens as drugs try to penetrate the cell is there are these membranes, these pumps that develop on the membrane of the cell, these efflux transporters, these efflux pumps. They're discovered about 35 or so, slightly over years ago. And what these pumps do is they pump the drug out. So you send the drug in, the pumps pump it out. Now, these pumps are not there all the time. They develop as a result of administering the drug or the cell observing that there is something bad that should pump out. So they would stay there for, you know, maybe 48 hours or something like that. And, you know, after that, they're gone. Now, you would say, well, maybe it's a great idea to neutralize the action of these pumps as a effective way of getting the drug into the cell. Well, you can do that, but that will create many other issues because these pumps are supposed to be there sometimes for getting rid of stuff that the cell really does not need. So it's not just drugs you try to administer to the cell. So that is an idea that was tried and did not work out. How can you fight these pumps? Well, you can administer a lot of drug. You can send a lot of drug into the cell, but then obviously you're dealing with toxicity. I mean, of course, eventually you'll get the drug in, but maybe you will need too much drug in order to get in. Okay, so the drug gets in and all sorts of things can happen to it. For example, it can be engulfed and metabolized, so there it's gone. Then it can, if this did not happen, it can penetrate the nucleus and maybe even damage the DNA. But, you know, a DNA is like, you know, have a lot of mechanisms like error correcting codes. So there are DNA repair mechanisms. So if you damage a DNA, it doesn't mean that the DNA cannot repair itself. So here, the effect of the drug is gone. Or maybe instead of that, the drug just gives an apoptotic signal to the cell. So signals that die, commit suicide. But there are anti-apoptotic mechanisms that kick in. And that's without even getting into all the environmental issues like hypoxia and so on and so forth. So in other words, when we think about drug, you have to have a very, very complex picture in mind of what can happen, what the drug is supposed to do, and what can go wrong. And when I'll speak about cells adapting to or avoiding drugs, it's not just a question of, oh, a cell mutated to be resistant to the drug and it no longer sees it. It's a very, very complex. And of course, you see from this caricature, much of which I did not describe, that, you know, you cannot capture everything that's going on. Okay, so why do we care about math and drug resistance? Well, several years back, we wrote a paper that was published in a journal, an experimental journal, really, Drug Resistance Updates, in which we tried to explain to clinicians and experimentalists why math can help them. it was actually a special issue for the discovery of these ABC efflux transporters. So all the other papers were like, you know, mice experiments and things like that. And then there was a paper about math. Now, it's definitely the most difficult paper I've ever written because there's not a single equation in it. It really is an attempt to explain what kind of questions can we attempt to solve using math. So there are several questions. I mean, these are a subset of the questions we presented there. For example, something we all understand well is an important problem, and that's what's the optimal protocol for drug scheduling in terms of dose and timing. When and how much should we give the drug? Of course, this turns into a control problem, and you know, you have constraints, like you want to minimize toxicity and so on and so forth. The way in which drug is administered in practice is really based on clinical trials, drug experiments that have nothing to do with, I would even risk and say very little, okay, maybe not nothing, very little to do with reality. Dosages are determined by taking a dish, putting a cancer cell line, figuring out how much drug you need to give in order for the cells to die, for half of the cell population to die after 72 hours. Why half of the cell population? Why 72 hours? I mean, that makes very little sense. Yeah, Tarot is encouraging. Yeah. Yeah, in this business, the more you learn, the less you want to know, really. So then question of continuous infusion versus short pulses. So typical chemotherapy, say a patient goes to the hospital or not, patient clinic, gets an infusion of a drug once every 3 weeks, once every 2 weeks, takes a couple of hours, goes back home. Is that the right thing to do? Is that the optimal thing to do? Maybe administering the drug continuously or doing something else is better. When several drugs are available, should you administer them all at once or sequentially, one after the other? This is a good question that mathematicians like to study, but doctors don't care about these days because they actually shifted the paradigm from thinking, well, in the past it was, well, I'll administer one drug, that will be my first line therapy. If that doesn't work, I'll administer a second drug, that's a second line therapy. These days, pretty much use a cocktail of drugs to target different mechanisms and hope that it works, even if you have nothing else to do if that fails. In many cases, when things fail, anyhow, the secondary drugs don't do better. So that, yeah, that is not very encouraging. Okay, so one of the themes that we'll see during the talk is about tumor heterogeneity. And the classical experiment goes as follows these days. They'll take a tissue, they'll pick several cells from the tissue, they'll sequence them, take cells from different locations, look at these arrows, and they'll look at the genome and say, well, the genome is different. So the conclusion, here's a paper, tumors are heterogeneous. Well, great, that is true, but we know that. What does it mean? Well, what it means different things to different people. And for us, during that talk, you should think about this, that if cells will have a heterogeneity in their ability to respond to drugs. So maybe there will be some cells that respond really well, some others with less likelihood. And there can be many, many reasons of why that's the case, but we will not get into these reasons. Just think about something that is going on here that provides some heterogeneity. And just to explain, and I think that's a very important point to make, that the genome is not everything. Here are a couple of pictures from experiments that were done by some collaborators of mine. Well, that's a Shelley Payton lab at UMass Amherst. That's part, well, AMIGOS is, that's an acronym that was coined by the National Cancer Institute. So it stands for, I mean, I think you would like it. It's Applied Mathematics and Germinating Oncology Solutions. So AMIGOS. So they have these different AMIGOS projects. And one of them I participated in which we studied plasticity, think about the response of different cells under different conditions. And the experiment here is an experiment where cells were grown in 3D gels. So that's an environment that's very close to what happens in the body. And the cells that are grown were these TNBC, that's triple negative breast cancer cells. Triple negative stands for, well, when these days when a patient is diagnosed with breast cancer, they look at on the molecular level at three things. Whether it's progesterone positive, if the progesterone receptor is positive or negative, estrogen receptor, and HER2. Positive, positive, negative, that's ER positive, PR positive, HER2 negative is what a doctor would call, an oncologist, good news. That's a common version. It can be relatively reasonably well treated. Patients, most of them recover. Triple negative, 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 that's the bad news. There are other versions. That's the one that they don't know how to treat, really, relatively poor prognosis. And that's why it's a cancer of interest. Each line here corresponds to a cell monitored over 48 hours. All these cells are identical. They all have the same genome. These are all identical. They're also next to each other. They have the same food, the same environment, the same everything. Whenever a line splits into two, that means a cell divided. Whenever you see an X, oops, that means that the cell died. So the picture is all over the spectrum. I mean, like, what's going on here? All these cells are supposed to do the same thing. They're all similar. They're all identical. They're all in the same environment. Why are they behaving differently? Well, we wish we could answer this question, but I'm putting this picture just to emphasize that genomic information is not everything. There's a lot of epigenetic stuff that's going on. That's how the cells respond to what's going on around them, even if it's all the same. Okay, so there's that. So in view of that, here is the like a vision slide. Okay, so in some sense, in that business, what will be the optimal goal? So the ultimate goal. So here are several assumptions. I'll call them as science fiction assumptions. And let me start with that. Let's think like a mathematician for a second, and let's assume that we know the initial data. So a tumor was diagnosed, and I can take, well, we can't do it, right? But let's assume, hypothetically speaking, that we can take the entire tumor, here's a picture in the bottom, and identify for each cell, if I'm going to treat it with some drug, how will it respond to it? Let's say that the yellow cells will die, the red cells will, the red cells will not die at all. The blue cells maybe will die, maybe not. Let's give them a probability of 50% of dying, and the green ones 75% or something. Think about heterogeneity in response to the drug. Let's say you can take the tumor and map it cell by cell. You know the whole picture. We can't do it, right? But let's say we know the initial data. Let's take it one step further, and let's assume we can fully control the treatment. We also can't do that, meaning we cannot say, I want my drug to go into this cell. We can administer, we infuse it into the bloodstream. You can target it in some other ways, but there's no magical way at the moment of saying, I want this drug to go there and that, how much. Let's say we could. And let's take it even one step further. Let's say we could dynamically monitor what's going on as a response to the drug, and we could adjust it. So that's something that we just mentioned in a conversation with Alex about his model. I mean, like, we wish we could do things dynamically in real time by monitoring. We cannot. Well, okay, let's assume we know everything. Here's my question. What's the optimal thing we can do, right? So what's the best one can do? So think about this for a second. What is the best one can do? So you have to define best, right? Like in any other, problem like that, it's ill-defined if you don't say what optimal means. For example, let's say I can get rid of all the yellow cells. Is that good? You tell me, yeah, it's good. You shrink the tumor. But then you have no more yellow cells. So when the tumor grows, let's say you have many more of the red cells, and you don't have the drug for the red cells. So was that a good thing to do or not? And maybe it's better to just get rid of half of the yellow cells. And now when the tumor grows, I'll have both red cells and yellow cells that grow. So maybe I can keep it under control for a longer period of time that way, kind of a chronic phase. Maybe that's a better thing to do. So I don't know. I mean, I'm just saying, here's the thing. And in a very fundamental question, and assuming that we know everything, we basically know nothing or close to nothing. And even that, you know, could we answer this question? Could we make sense out of it? So I'll not answer this question today, but I'll give you some steps about how I'm thinking about this problem and what kind of steps one will have to take in order to get there. As I said, drug resistance in math did not start, the study of drug resistance in math, or math and drug resistance, did not start with cancer studies. It actually dates back to the work of Luria and Delbrück in 1943, for which they won the Nobel Prize in Medicine in 1969. And that's, in some sense, a Nobel Prize for a mathematical work. It's mathematical and experimental. I'll actually explain to you the experiment. I'll show you that, not so much the math. Well, I want to speak about different kinds of math, but I think that's a good opportunity to mention this experiment if you've never seen it. So in modern language, the question that we'll ask is a question about antibiotics, bacterial resistance to antibiotics. At the time, that was not the question. This is prior to antibiotics. So the question that they asked was, if you take a bacteria and a bacterial culture and you challenge it with a phage virus, think about something that kills a bacteria. Oh, how about the antibiotics? Most of the bacteria is killed. But then not everything is killed, okay? When you take a culture, put bacteria and you put antibiotics, not everything is killed. What happens when you let it then grow? Well, the observation was that if you then use again the same phage virus, these cultures don't respond. They develop resistance to the virus. And the main question is, where did this resistance come from? That's a question that they wanted to answer then. Nothing to do with cancer, but still very interesting, right? So think about this as two options. Option one is resistance is natural selection, meaning what? Well, you started with bacteria. Some of them may have been already resistant to this drug, this phage virus, before you administered it. So what you did was you killed everything else. Remember these yellow cells? You kill the yellow cells, you pick the red, they grew. You can't do anything with them now. That was option one. Option two, the resistance is acquired. So what you should think of is like that there is some mechanism that bacteria are very simple creatures, if you wish, right? Cellular organisms, single cell organisms, but they still may be more sophisticated than we think. So maybe there is something then that for whatever reason, these bacteria did not die. It's not as if they were resistant up front, but they did not die for whatever reason. And then those that survived did change something in them. Some mechanistic thing happened that blocks infection. And then when you challenge them again, I can't do anything against them. So think for a second, I mean, how on earth would you tell the difference between the source of resistance? How would you know? Because the outcome is the same regardless. I mean, at the experiment, you started with bacteria, you put some drug, most of them were killed, some stayed. Then they grow, you try again the drug, it doesn't work. How would you know where that came from? Think 1943. So that's basically the work of Luria and Delbrick. What they did was they designed an experiment, and then they did the mathematical analysis of it. And the experiment is quite clever. So think about the following experiment. Take very small pieces, take very small samples of the culture. So it's so small that you assume really there's nothing there. There will be no resistant cells. Now you may be wrong, but let's assume that that is the case. And after that, what you do, you expand them into a fixed size. So you do that. So you expand them, expand. Now, what happens is that after partial growth, there's no drug yet, or this phage virus. It's not there. So maybe some cells develop resistance to the drug, even though there's no drug, okay? So that's this picture here. This picture, nothing happens. You keep growing, nothing happens. In this case, if that happened, then what you see then is maybe in one of the plates there is nothing that's resistant, red is resistant, and in some others there'll be, oh, so you see there's a high variance in what you would see in terms of these, in terms of the number of cells that are resistant to the drug. Now you administer the drug in, and then you do the whole experiment, right? So you let the things grow and you administer it again. Basically, you see a difference between these two settings with that experiment. If that was what's going on, and the resistance developed only as a result of the drug, then you should see a similar picture in every dish, more or less. While if resistance was the natural selection thing that's not the result of the drug, then you should see a high variance. You should see dishes with different types of numbers of cells. Okay. So they did this experiment, and they also did the math. And where does the math come into play? The math comes into play because you can estimate from the variance the mutation rate. And you can say something clever about that. And it's actually very interesting. The original paper is also very, very interesting. It's, you know, a 1943 paper. So if you have some time, I highly recommend to look at it. It's one of these things that were written when still math was written as if it was poetry. So it's very, you know, it's really nice. And the math is interesting. The result, by the way, is that it is natural selection. The resistance did not develop as a result of the drug. So it is something that's there all the time. And you can think about cells that mutate in the background. It doesn't mean, of course, that these processes are mutually exclusive. So you can think about, you know, when the doctor tells you, or when you read about, you know, don't keep taking antibiotics so that you don't develop drug resistance to the antibiotics, then, I mean, that is not the natural selection. They're thinking about that. So this can be a combination of these things. But the major process that's happening here, first approximation, is natural selection. Okay. So I tried to avoid math, and now I'm jumping into, you know, I can call it a puddle. But in order to do that, after trying to convince you that this is a super complex problem, let me go to the other side of the spectrum and show you what is the simplest possible thing you can do if you were to go now and write a model for drug resistance. What's the absolutely simplest thing you can do? And then I'll gradually make it more and more complex. So the simplest possible thing you can do in my mind is the following. First, I assume everything deterministic. Let me not assume any stochasticity here. Then I will assume only two cell populations. I have these yellow cells. The yellow cells, I don't know why my green cells are green and not red, but in any event, the yellow cells are sensitive. These are the wild type. I'll call them N. These are the natural cells and wild type. These cells, once I administer the drug, they will respond to the drug. Then I have resistant cells. These, in this case, will be green. These are the capital R. They, when I administer the drug, they will not respond to the drug. So I have time T star will denote the time by which the tumor is diagnosed. I start treating the tumor. And what happens before that? Linear ODs. Can't be any simpler than that. Linear ODs. The wild type cells, the sensitive cells, before being treated, they grow exponentially. I can put here anything. I said the simplest possible thing, exponential growth. L is birth rate, D is the death rate. So you have an effective growth rate of L minus D. Of course, I assume that L is greater than D. And yes, cancer cells die. So I'm putting a D here. What about the resistant cells? Simplest possible thing. So I don't assume anything about their growth rates being any different than the sensitive cells. So let's assume that L and D are the same, so they also have this exponential growth. And I'm also assuming that there is certain influx of these cells. So there's a mutation rate here of U, and I'm taking N and converting them into R. Now I'm not subtracting it from N, because think about, I'm just scaling things in a way that, think about a cell dividing, and when a cell divides, it creates two offsprings, and when these offsprings are there, one mutated and the other one did not. So one became resistant and the other one just stayed in the population of the wild type cells did not change. So I'm not subtracting it from that other equation. By the way, if you ask an experimentalist, do resistant cells proliferate faster or slower than wild type cells, than sensitive cells? The answer is it's completely inconsistent. Sometimes they will be slower to act. Sometimes they're faster. By the way, faster in general is better in some sense, but not in the case of resistant. Now we can come to that later. Okay, after T star, what changed? Nothing with the resistant cells, and with the natural cells, I just kill them with a higher death rate, okay? So that's that. Again, simplest possible thing, linearly, you can solve them explicitly. These are linear equations, nothing fancy. Well, even though we can solve them explicitly, it's a little bit more convenient not to, just to be able to see what's going on. And you can start asking questions. For example, the total number of cells, if you call that M at the time of detection, if you call M the total number of cells at the time of detection, and naught would be the number of cells at time 0, you can represent the time of treatment as a function of these parameters, and you can estimate how many resistant cells you have at the beginning of the treatment. Okay, and that expression will involve the mutation rate, the total number of cells, what you started with, and the birth and death rates. In fact, if you went to go and say, well, why one drug? Let's put 2 drugs. So I have resistance to drug 1, resistance to drug 2, resistance to both drugs before the treatment, and let's say that everything responds in the same way to the drug other than those that are resistant to the both drugs. They can't respond, these cells. It's actually the number of cells that you start with is power, but basically very similar expression to what I had before. And then from that point, you can just a power of 2, put power of P for the number of drugs. What is then interesting, where can you take such a model to? You can say, well, I can measure how many cells I have before the treatment, resistant cells, and how many I have during the treatment, and which one is a bigger population. And if you do that calculation under reasonable mathematical assumptions, you get that the pre-treatment resistant cells is a bigger population than the during treatment resistant cells, and so on and so forth. This is really like, stretching the linear differential equations as much as I can. So I said it's as much as I can, so I'll try to do something a little bit more complex. And the next thing that I'll do to make things a little bit more complicated and say, well, I said, here's a population of, in the previous model, I had sensitive cells and resistant cells. It was a black-white thing. I tried to convince you that there is heterogeneity, and the heterogeneity is something that is not black and white. So let me do something that's convenient to do mathematically, although one can argue that there is a biological meaning to it, and I'll not address it here. And that is that resistance level in the next models will be assumed to be a continuous parameter. So think 0 are my sensitive cells, 1 are fully resistant, but I have all the spectrum in between. And I'll show you how that will impact the model. That's an idea that started by a French group, so Benoit Pretam and co-authors. Tommaso Lorenzi, I think at the time, was associated with him. Now he's in Torino, okay? In any event, here's the kind of model I want to write. So I'm going to take N to be the cancer cell population, and I want to follow its evolution over time, only that I have here 2 parameters. That's a bit confusing. I'm using X. X is not space yet. We will get to X being space. But in this model, X is not space. X is my trait. It's a level of resistance. So as I said, 0 is sensitive, 1 is fully resistant. And think about the continuum of things in between. So what can these cells die? What can the cells do? Well, one thing they can do is they can they can proliferate. So I have a proliferation term. That's this R thing. The proliferation can depend, in principle, on the level of resistance. Maybe in some system, the cells proliferate slower when they're resistant. Maybe they're faster. That allows me to model that. What else do they do? Well, they die naturally. Again, that can be a function of the resistance level. What else can they do? Well, I'm adding here a drug, so that will be another thing that will kill these cells. What else can they do? Well, they can mutate. So I'm allowing a fraction theta of x, cells with resistance level x, to mutate. So I'm removing them. And then, well, this is the source term for the x. So take all the other ones, the y's, take all these cells that had y's and allow them to become cells with x as resistance level. So I'm increasing here the population of cells with trait resistance x. There's another thing that's hidden in this model, and there's a kernel here. And I'll start by assuming that it is symmetric. It's kind of a dumb assumption because, like, cells that mutate usually do not mutate back. They exist back mutation, but that's a very rare thing. But you can put here any mutation kernels that you want. I'll show you something non-symmetric. In any event, there's another thing that's hidden here, and that's a density. Now, density is not very meaningful if I don't have space. So think about me taking a time t and just counting how many, the total number of cells I have. I will convert it into something that has space later. But what I'm doing by it, I'm trying to have here some mechanism by which, if there are few cells in the system, they can behave in a certain way. If there are many cells, if the system is saturated, they can behave differently, and anything in between. So I'm adding such flexibility to the model. Now, of course, the way it is written, it's like thinking about all the mechanisms, then you start realizing that there's a lot of redundancy here. So you do the simplification and you scale time and all that stuff. And the same model, you can simplify and write it like that. The change in the cell population, there's a birth term, there's a drug term, there's a death term, there's a mutation term, and everything that has to do with the fake density I'm throwing onto this term. Okay. And now what I want to do is to understand how this equation behaves, how solutions to this equation behave. So I'll do it in 3 steps. The first step is I'm going to strip off the mutations and the density. So all that I'm left is with an equation that looks like that. I'll stare at it for a second. If I fix x, this is just exponential growth, or n will exponentially increase or decrease, right? I mean, what can happen here? That's it. Okay. Then I'll have the density. I'll add the density, and I'll ask, how does that change the dynamics? And finally, I'll add the mutation. So what happens here? Well, the first case, which is nothing but something that looks like an ODE, although really a system of ODEs, or a continuum of ODEs, because X is a continuum parameter. What you can show is that asymptotically, well, of course, the total number of n, I mean, you go that exponentially, that's an exponentially increasing function, right? So the total number of cells, just goes to infinity, right? Of course, it depends what kind of functions I put here, but put something, okay? The death is very low, and then say something about the growth and the death as a result of the drug. The natural death is also always very low. Okay, so the total number of cells will go to infinity, but if you normalize by the total number of cells, that becomes a little bit more interesting, and that turns to be just a collection of delta functions. So asymptotically, what you have is a selection process that peaks in places where you say, well, that is a trait that I'll have. 0 fully sensitive, 1 fully resistant. So here I have something with 0.58, whatever that means. Okay, so that's a symptomatic peak over there. Okay, now I'm adding the density. What happens if you add the density dependence? Of course, depends on how you add it, but under reasonable assumptions about the behavior of this term, it turns out that the total number of cells here is bounded. So the density remains bounded. That's a theorem. But when you look individually at the cells in different x's, you still get this behavior of delta function. So the limit distribution really is a collection of Dirac masses. Well, if you add the mutation, the density is still bounded, but you no longer get these delta functions. And what you get is these distributions that spread in the X domain. And the spread is something, so you have here like, you know, an actual variance here, a non-zero variant. The spread is actually something that you'll interpret as heterogeneity in that profile. So you have cells with resistance level all the way from here to here in the black curve. Okay, so not just one thing. There's a tumor that has many kinds of heterogeneity. Okay, so that's it. Well, this is nice, but that model is not something that has any space in it, right? So let me add space to the picture. And when I add space to the picture, the first thing that I do, of course, is I add a radially symmetric model. So there is a model, the same group wrote a model that was radially symmetric and included some basic ingredients. What are these ingredients? The cancer cells grow, they die as a result of natural death, and they also die as a result of a drug. In this case, the notation R is the space dimension, that's a radial distance. Theta is what X was previously, so that's just a level of resistance. Okay. There are two kinds of drugs in this model. One is a cytotoxic drug that kills it, and the other is something we did not have previously, that's a cytostatic drug. That is a drug that slows down the growth of the cells. R is a growth. It slows down the growth. The growth is a function of the nutrients that the cell gets. So the nutrients are something that you diffuse into the system, you can consume, you can add the source of them, and then you add equations for the drugs. Same thing, there's a source of the drug, they diffuse, they're consumed. In the spirit of what we learned from the other model, we took that and I would say improved that model and allowed some things to happen. First, we allowed cells to diffuse. They are not stick to their R, to their band where they are as they are in the previous model. They can move along the tumor. That's its diffusion. We also allow mutations, and there is the density dependent. So that's a radially symmetric model. You can say all sorts of things about the uptake functions, how the drugs are consumed, how the nutrient is consumed. Let me ignore that. Let me show you a picture. And that picture is interesting. Let me focus on the center picture, the one in the middle. What you see here is a picture over time. This is a radial model, radially symmetric model. I am looking now at the distribution over the entire tumor of the level of resistance of the cells. This axis is the level of resistance, fully resistant, fully sensitive. I'm starting with cells that have about, around 0.5. This color scheme just shows you where's the concentration of cells here and which level of resistance. And what you see in this picture is that they concentrate over time around this value. We are administering a certain level of the cytotoxic drug, the one that kills these cells. What happens if you increase the level of drug? What happens then is two things. One is you see a period without any cancer cells. The cancer cell population goes down. This is what one would call a remission. I don't see cancer. But then there is a relapse. The cancer comes back. And when it comes back in this model, it comes back at resistance levels that are much higher. If I further increase the population of the drug, the dose of the drug, I get a longer period of without seeing the cancer. But again, when it comes back, even higher resistance. I can try it with an on-off scheme. And in fact, if you pay attention to what happens, and of course, the dosage are set so that the dosage over an interval here just administered in a shorter interval. That's like administering a higher dosage, and that will actually, again, delay the relapse, but will cause higher resistance. So this is worse than that, at least in this model. Now you can study the role of different parameters. Alex, how much time do I have? I want to be...
So typically it's until 4:30 with questions, including the time for questions.
Okay, so let me skip these cute pictures and show you another interesting pictures. Here is not time, but that's R, the distance from the center of the tumor, the very center, and that's the membrane. And that's a level of resistance. So what you see here is that under certain conditions, what emerges is a heterogeneous tumor. There is a region from here to, or here, let's look at this here, from here to here with a certain level of resistance, and there's a different region close to the membrane with a higher resistance level of cells. So this basically corresponds to something that I have in mind here, if that was a real 2D model. Okay, so if you don't like symmetric kernels, you can put them asymmetric, so these distributions shift. If you think that that's continuous stuff is not really nice, so you'd rather have some points with some jumps between these states, you can also do that. It doesn't really change the results. It does make these curves kind of ugly. But that was radially symmetric, so can you do the real 2D? Well, yeah, you can. You need for that to pick a model for, I mean, technically what I'm writing is cancer growth and death use of the drug and diffusion. All that stuff is good. You know, cells can move. What happens is that you then have to, that's more complex because you have to decide what to do with a growth model. The growth model, you have to take something from a, you know, there's so many cancer growth models out there. So what we do here is something to think about a pressure that comes from the inside of the cell, and somehow you identify the membrane as a kata function, and you keep things under a homeostatic state. And this is not bad. You do that, but everything else is the same. Now, 2D is not symmetric. So now when you administer a drug, you have to decide whether you administer it symmetrically around the tumor or from the top or with some fictitious vasculature. This doesn't really mean anything other than blue is areas where I can give nutrients and drugs, and red I cannot. And one thing that you then do is you decide if your cancer growth model is good or not. And it's actually cute because what you see as the tumor grows is the center of the tumor dies off, and that's what is known as necrotic core. The cells in the center of the tumor die, and only the centers on the outside of the tumor proliferate. So that's actually what you expect to see. You can even do some math and say something about the propagation of the boundary. But what I want to show you is this picture. And this picture is a picture where I start with a toy problem of blue cells being sensitive and red cells being resistant. And I administer the drug from above, okay, from the top of the domain. And I'm staring at this tumor from above. What you're seeing here is the density of the tumor, and what you're seeing here is the level of resistance of the cells in the tumor. So no drug. What happens? Times 0, times 10, times 20, the tumor grows. It grows towards that edge because that's where the food comes from. Now I put some drug. What happens? I have a smaller tumor. But look at what this tumor looks like. It's mostly resistant cells. I put a higher drug. Everything looks resistant. I don't see it, but I wait longer, it will come back. Okay. So which one is better, this one or that one? Well, similar picture, just a different initial data. So say here are pieces, okay? Which one is better, this one or that one? Well, that's to decide, right? Heterogeneity develops. You can do the administration, as I said, you don't have to do everything uniform. So you get like really cute, boundaries. By the way, this is how boundaries of cancer cells really look like. Sometimes nothing special. Sometimes it looks like these flames, kind of like that. But this is really doesn't mean anything biologically, but it's still interesting that it will look like that. Sometimes there's this collective behavior that it's as if you're sending like things outside. And actually the worst one is, no one to see that these individual cells that are lingering them and who knows where they go next. So that's that. So, I mean, we are kind of very running out of time. So I'm not going to do the optimal stuff, but let me say some things about that. Okay. Mathematical oncology develops in many different directions. One direction that had developed extensively is tumor growth. Lots of models out there. Another direction is drug resistance. Lots of models out there. Why don't you see models of both? Well, these are models of both, but why aren't there so many of them? You know why? A curse of dimensionality always goes back to that problem. Remember the previous model I showed you, 2D in space? Another variable for resistance plus time, 3 + 1. If I want to have many drugs and the 3D in space, I'm kind of computationally stuck. Well, okay, then you have to come up with clever computational ideas, and this is a different talk. The other thing that's a problem, let's say I have here several cytotoxic drugs. They can all kill the tumor. Several cytostatic drugs. They can all slow down the growth. And I administer all of them at the same time. Let's say that I even hypothetically know how to do that computationally. But what I don't know, what I'm missing is data about the cross-reactivity of these drugs. I need an experimentalist to tell me that. If they tell me how these drugs act together, I will change this term to represent whatever is the actual experimental action of these drugs. But same thing for here. But I mean, in absence of these here, what I'm writing here is you give me capital M drugs that slow the tumor, just add all their actions together. Of course, that's not going to happen. It's capital M drugs that kill the tumor. Well, they all kill independently of each other. Well, that's how it's written here. That's not what happens. But in spite of all these experimental groups I've spoken with, I could never find data on this cross-action between drugs. So as I said, there are computationally interesting questions one can ask about that. And there are ways of simulating things between drugs, high-dimensional models, but again, that's not this talk. And the very final thing that I wanted to say is that everything that I did was in vacuum, if you notice, like, there are cancer cells and there are drugs. Okay, there's nutrients, but where are the healthy cells? Where are the normal cells? So one can actually model competition between healthy cells and cancer cells and do it in the framework of these models. It's kind of difficult computation. No, well, not so difficult computation. I should say what is difficult here is to argue why that is exactly relevant. So here's the thing, like, when you see cancer models, cancer growth models on discrete models on a grid, so they're like rules, right? So cells move and when they can proliferate and die. When do they proliferate? They proliferate, according to these models, they can only do it when they have space around them, right? I mean, that's how it works. When there's a neighboring site that has space. Cancer cells don't work that way. Cells in general don't work that way. When they want to proliferate, they proliferate. And it'll push things away. So, I mean, on the continuum level, it's actually easier to model than if you were to write now a discrete model to describe something that looks more like that than just, a motion on a grid. But you can do that competition models. I actually don't have anything intelligent to say on that other than you can do it. So you can model the dynamics over time. The blue is healthy, red is resistant, and under certain conditions, basically you get what you expect to see. Okay, so with this, I'd just like to summarize the talk and say that my goal was to give you some ideas about how math and drug resistance and cancer can, be used or, work together. I think that there are a lot of problems that we do not know how to solve. There are multiple specific cases with specific drugs and things like that we tried, but it's not as if I can give you any particularly new insight when it comes to these models. So I set these examples aside, but I encourage you to think about some of these problems. And, in terms of the math, what I like is that, not only the new challenges, but maybe one day that way of thinking will also be useful. So with this, I'd like to thank you for your attention, end here, and take any questions. Thank you so much.
I'm going to clap in person for everyone involved. So thank you so much for giving the thought-provoking talk, telling us what models are, why they are that way, and why they should be in different way, what we can and what we should strive for. Are there any questions? Please just unmute yourself. Go for it.
So I'll ask a question. This is John Guggenheimer. There's a tremendous amount of detail, particularly in the last things that you were talking about. And you would like to make models that sort of target the important details and ignore the others that in some sense reduce the complexity of the problem. What sort of strategies are people using in studying cancer to do that?
Well, this is an excellent question, and really things go in opposite directions. So when I give a talk, and I give a talk in front of an audience that has half medical doctors and half mathematicians, and if I put a model that has 4 parameters in it, half of the audience thinks that this is too simple. The other half thinks that it is too complex. And let's see if you can all guess which one thinks it's too simple and which thinks it's too complex. As mathematicians, we want to get less and less parameters. I mean, for me, the biggest challenge is usually to convince people on the experimental and or clinical side that some things should be left out. Okay, so look at them and say, oh, where's all that stuff? Well, so we can't have this all. But then I go back to them and I say, do you have any intuition? Well, here, 3 different kinds of cells. I mean, do I really need all of them in the model? And then they think, and sometimes they come back and say, well, you know what? Actually, the regulatory T cells should not be there because, on that time scale, they will not play any role or something like that. So really, in other words, the simplification in many cases goes back to the actual biological problem. And what you could not see in this talk is I'm talking about some principles, and I'm writing these models that work on principles, but I didn't go to exactly, as you say, with this talk to these examples where I said, well, I can't do that. So now I have to start, thinking about how to simplify the model. But that usually comes when I will look on a particular disease in a particular setting, because these models, another thing I didn't say, these generic models, we pretend that they're good for everything. At the end of the day, they probably are good for nothing. And to make sense out of them, you have to really adopt them to data and to make them useful. With this being said, there is the other school of thought that, you know, sometimes people that are that have some criticism against with regards to models that have, I don't know what, you know, dozens of parameters, use approaches that can be very important approaches and very useful for certain problems, but maybe like, you know, could be machine learning, for instance, where I take 17 million equations, none of which has a biological meaning, and we believe the result of that, just because we managed to, you know, fit the data and validate it, whatever that means, and do something with it. So it's a constant struggle. I mean, it is a constant struggle. I remember once we wrote a paper on the action of the immune response, and the contraction of the immune response after a primary viral infection. So the question is, what causes the immune response? After you get a virus, you know, the immune response normally kicks in and then it comes down. What causes it to come down? That was the question. And we wrote a very simple model that demonstrated it beautifully with the wrong biology, of course. So we sent it out for publication. We get back the reviews. Reviews liked it, other than one reviewer that said, nice paper, wrong biology. So yeah, I know. I mean, we assume that they're regulatory cells that come from CD8 cells. They actually come from CD4 cells, but well, so we said, well, what can we do? So we kept the paper as is. We took the model that was a very simple model that explained precisely the mechanism that we wanted to demonstrate. We tripled it in size and the number of equations, added the right, converted it to the correct biology, hid everything. I mean, everything at that point was hidden inside in terms of the mechanism, and put it in an appendix, ran a couple of simulations, said it's the same. We send it out, the reviews come back. I said, well, you know, it's nice. I kind of believe you, but why did you pick only 2 of your examples? Well, so we reran the entire big model and everything. At that point, it no longer fit in an appendix. We had to have a massive chapter that explained everything, but essentially the results were the same. We send it out. The review comes back and says, well, if the results of both models are the same, why do you need the first model? So we ended up publishing a paper that had nothing to do with the paper that we sent up to begin with, just because of this going in the reverse trajectory. And later I published a paper saying a simple model of where I actually said what I wanted to say. So there is no good recipe for how to do it. I think that the value of very simple models is super important. And I think that complex models are very nice when you get them to do what you want them to do. And that is not an easy task. If you write a mathematical model that has many equations and you use parameters that are within the biological range, acceptable biological range of these parameters, and you get results that look the way you want them to, I wouldn't call it, I actually don't take a negative approach of like, oh, the model is so complex you could get anything with it. I take a positive approach. I'm saying the model is so complex, the dimensionality, the search space is so huge, and I still see what I wanted to see. I should feel lucky. So.
Thank you.
Okay. Since we ran over time, I'd like to offer our thanks to the speaker again. Let's applaud once more. And those who want to stick around and continue the conversation are definitely welcome to do so. So thank you, Doron.
Thank you so much.
We always, when we invite people to give these talks, we ask them to give a general introduction to research area, keeping in mind how broad are the profiles of people in the audience. And it's very hard to resist the temptation to talk about your latest and greatest to the detriment of introduction to the area. So thank you for resisting the temptation and for giving a very good intro in the first half.
Yeah, I mean, like, You know, this is maybe a comment to the students in the audience that they can make, like, as, for example, when I was a postdoc and started going around and giving talks, what I used to do is, you know, if I would give, I don't know, like the first year I was a postdoc, I gave several talks, and it was very important for me to, every talk I give, to have it on a different topic. Like as if everyone that attended my, that attends this kind of talk already heard me talking, you know, previously. So yeah, and then I, you know, over the years I realized, one, that's not the case. Second, I mean, it's kind of like, it's not very effective to walk into a place and say, well, last time I've been here 5 years, that's the point I stopped at, and let's go on from there. And the third thing is that, you know, in any discipline you're looking at, there's so many interesting things that happen, and to necessarily pretend that, you know, you invented everything is not very instructive. So kind of like part of what you say should be based on why at all you're looking at these kinds of problems. And, you know, so I like the more expository talks, and, you know, it's okay to have some technical details, but, you know, if someone is interested in any of that stuff, you know, that's what we invented journals for. And so
I have a different question about your experiences working in this area. Many years ago, I was on a panel for NIH that was evaluating proposals for centers for computational cancer biology. And I was hardly impressed with the level of mathematical sophistication and the extent to which I had the feeling that really innovative mathematics was suggested being used in the proposed centers. So the question that I have is that over the time that you've been involved in this research, have you seen a significant change in the receptivity and attitudes of the cancer biologists to different kinds of mathematics? And on the other hand, a significant increase in the extent to which mathematicians have gotten deeply involved in the cancer biology and been able to make good contributions.
yeah, so these are two interesting yet related questions. I would say that first, you're absolutely right that things at times can be quite rudimentary, not to say that sometimes all that you need in order to solve a scientific problem is rudimentary analysis, but nevertheless, there are some fundamental things that should be thought of when it comes to such, you know, trajectories. So one is on the level of the mathematical community. One thing that I noticed is that when you look at mathematical biology, in the past several years, there have been a tremendous shift from the classical problems of mathematical biology to math and medicine in general, not just math and cancer, but mathematical medicine, I'll say that in general. So if you go to, I don't know what, like an annual meeting of the Society of Mathematical Biology, many of the sessions are related to medical areas. So I view that as opposed to like the past where, you know, that kind of like would move from modeling shrimps to modeling calamari or this pattern or that, but which can be super interesting problems, but it was more traditional mathematical biology. So I do see a positive thing that's happening there. And so that's one thing. The other thing, in terms of the openness of, you're asking the openness of the, on the medical side. So I do see this, things improving in various directions. I do view that as an improvement. One is that there are several hospitals that have mathematicians in them these days. So examples for that, examples are Sloan Kettering, Moffitt Cancer Research Center has a mathematical oncology department. So Sandy Anderson, David Basanta, Heiko Enderling are people that are there. The City of Hope Cancer Center in next to in LA, next to Pasadena, has also a mathematical oncology group. So gradually it trickles into the hospitals, which I think is very, very positive. And when I say math, I mean math and not bioinformatics and not biostatistics, actual math and mathematical modeling. So that is positive. On the same note, I had the fortune over the years to communicate with many of the branch chiefs and laboratory directors at the National Cancer Institute. There are about 50 different branches. And I'd say that in terms of their attitude to math, you can divide it into three groups. One is those that think that it's completely irrelevant. I accept that. Could be irrelevant to what they're doing. Another group thinks it's relevant and like the idea, and then you speak with them and it turns out that that's bioinformatics. They call it math, but it's bioinformatics. And the third, and it exists, a group of people that see the difference and understand that mathematical modeling is different than bioinformatics. And sometimes the way that they get to it is through peripheral areas, like, they have images and say, well, imaging, we want to do stuff with these images. And they realize that that's not bioinformatics, but what kind of so is that computational? Is that, so it comes from a different direction. And all of a sudden that leads to other approaches. A downside of what's happening, because you mentioned the study section there, is there's only one study section in the National Institutes of Health, the MAPS, the acronym is Modeling and Analysis of Biological Systems. It's the only one that under its mission lists math. And that is periodically controlled by people that are not mathematicians. In fact, I don't think it was ever controlled by mathematicians. So sometimes it's a computer scientist, some other times it's a bioinformatician, a bioengineer, and then it goes back to the computer scientist. There are mathematicians, real mathematicians that we know that sit on that study section, but they're a minority. So there is an issue. So even though there is an acknowledgement that this is important, we do not see enough funding and enough initiatives that go in the direction of math directly coming from the NIH. It's still a weird kind of a thing there, but there is hope. And these centers, there were these PSOCs, physical science ecology centers, and the integrated cancer biology centers, these two types of centers. And the first one was taken over by physicists and the second one by bioinformaticians. So mathematicians are not really big players, very minor in most of these.
Thanks.
There is hope though, sure.
Doron, thank you again. I will have to disappear right now. I hear my family on the other side of the door.
Well, thank you so much, all of you. I mean, it's been a pleasure, and yeah, hopefully one of these days. Are you all fully vaccinated?
Some people are at different stages.
Okay, so yeah, one of these days, yes. Here or there, yeah.
Indeed. Very nice to see you as always.
Pleasure.
My best regards to everyone in Maryland.
The same day.
Bye-bye.
Thank you all. Take care.